Hi everyone. Here's our next free response question. Now this one was also from the 2019 AP Bio exam, and this was question 1. Now this is not an experimental design question, but I think it's a really good example of a concept explanation question. Remember, on this year's exam you're going to have 2 free response questions. One's going to be on experimental design. One's going to be on concept explanation. Now, this question I'm about to go over with you is about more than just concept explanation, but it's got some valuable stuff in there I think it's worth looking at. So one of the things the College Board will do is ask you to interpret diagrams. So let's take a look at the diagram for this question. It says you have gene TRPT gets transcribed into mRNA. Well, they don't tell you transcription, but you guys all know if you go from a gene to mRNA, this is transcription. Okay. And then the mRNA goes to enzyme TRPT, and that would be translation. Okay. And then on the other side of the diagram, it says gene YUC gets transcribed into mRNA, and mRNA gets translated into enzyme YUC. Then, if you look across the bottom of the diagram, it says you start with tryptophan. Enzyme TRPT catalyzes the chemical reaction that turns tryptophan into I3PA, and then I3PA gets turned into something called IAA, and that reaction is catalyzed by the enzyme called YUC. So what this is showing is a model of a 2-step enzymatic pathway of how you make IAA from tryptophan. Now, to answer this question, you do not need to know what tryptophan is or I3PA or IAA. Okay, so that's the diagram. They're showing you this gene codes for that mRNA, which gets translated into this enzyme, and this enzyme is necessary for the first step in this pathway. And then this gene gets transcribed and then translated into a different enzyme, which is needed for the second step in this pathway. All right? Auxins are plant hormones that coordinate several aspects of root growth and development. Indole-3 acetic acid, IAA, that's this guy, is an auxin that is usually synthesized from the amino acid tryptophan, figure 1. Gene TRPT encodes an enzyme that converts tryptophan to I3PA indole-3 pyruvic acid, which is then converted to IAA by an enzyme that is encoded by the gene YUC. So they're giving you a lot of background information in this question. So please do not be afraid or freak out or freeze up on the AP exam if you see something on there you've never seen before in your life, like IAA. Okay, so look at part A. Circle one arrow that represents transcription on the template pathway. Identify the molecule that would be absent if enzyme YUC is non-functional. So there's 1, 2, 3, 4, 5, 6 arrows in this diagram. They're asking you to circle one that represents transcription on the template pathway. So you could have circled this arrow or that arrow, because both of those are transcription, because transcription is going from gene to mRNA. Okay? Identify the molecule that would be absent if the enzyme YUC is non-functional. Okay, if enzyme YUC is non-functional, that means that step can't happen. Okay? So what molecule would be absent? Well, that molecule would be absent, right? IAA would not exist. Okay? B. Predict how the deletion of one base pair in the 4th codon of the coding region of gene TRPT would most likely affect the production of IAA and justify your prediction. Okay, so if you delete one base pair, okay, what kind of mutation is that? If you delete just one base pair, that's a frame shift. And remember, frame shifts are usually more severe because that frame shift, that deletion of one base pair, is going to mess up that codon, but it's also going to mess up every codon after that. So if you do a deletion in the 4th codon of the gene TRPT, that means that enzyme would be non-functional, right? It'd be really messed up. So that would be your prediction. Your prediction would be that you wouldn't be able to make I3PA or IAA. And how would you justify your prediction? You would say the deletion of that one base pair is a frame shift. That would probably make this enzyme non-functional, so you wouldn't get past tryptophan. Hope that makes sense. C, explain one feedback mechanism by which a cell could prevent production of too much IAA without limiting I3PA production. So this is kind of like the tripoperon we learned about, where the tripoperon is in charge of making the enzymes that produce tryptophan. And if you remember, once the cell has enough tryptophan, tryptophan goes back and shuts the process down. So a feedback mechanism that would prevent too much IAA would be if IAA shut down one of the enzymes in this pathway. But the question's asking without limiting I3PA. So if IAA shut down this enzyme, then you would also be shutting down the production of I3PA. So the correct answer would be IAA shuts down this enzyme, enzyme YUC. Now, part D and C really have to do with ecology, so we're not going to go into a ton of detail on those, but let's take a look at these really quickly. Rhizobacteria are a group of bacteria that live in nodules on plant roots. Rhizobacteria can produce IAA and convert atmospheric nitrogen into forms that can be used by plants. Plants release carbon-containing molecules into the nodules. Identify the most likely ecological relationship between plants and rhizobacteria. Okay, so rhizobacteria convert nitrogen into forms that can be used by plants. So the rhizobacteria helping the plants. The plants give carbon to the bacteria. So the plants are helping the bacteria. What do we call the kind of symbiotic relationship when both benefit? You may remember this from integrated science, but that's called mutualism. Mutualism is when both members benefit. So identify the most likely ecological relationship, it would be mutualism. Describe one advantage to the bacteria of producing IAA. Okay, so when the rhizobacteria produce IAA, how does that help the bacteria? Bacteria don't need IAA. What does IAA do? Okay, well, if you go back to the beginning, the stem of the question, IAA stimulates the growth of plants. Okay, so how do rhizobacteria benefit? Well, by producing IAA, they're stimulating the growth of the plant that they're getting their food from. Okay, now in part E, this was really kind of a funny question, I thought. A researcher removed a plant nodule and identified several cheater bacteria that do not produce IAA or fix nitrogen. Okay, so they're kind of like parasites on the plant or something. Describe the evolutionary advantage of being a bacterial cheater in a population composed of predominantly non-cheater bacteria. So what would be the advantage of that? Well, if they're cheater bacteria, they would be getting food from the plant essentially for free without having to do anything in return. Plants can adjust the amount of carbon-containing molecules released into nodules in response to the amount of nitrogen fixed in the nodule. Predict the changes in the bacterial population that would cause the plant to reduce the amount of carbon-containing molecules provided to the nodule. Okay, so if there was too many cheaters, then the cheater bacteria, then the plant would reduce the amount of carbon it would produce. It would give back to the bacteria. Anyway, hope that makes sense. Now, in our next video, I'm going to go over what the scoring guide was for this question, and we'll look at an example of a really good response to this. Bye for now.